Okay, so ladies and gentlemen, welcome. So in today's lecture, we are going to look at the structure of DNA and the different types of DNA. So introduction to DNA structure. DNA is a DNA is a vital molecule carrying genetics information in living organisms. So all the genetic characters they are actually carried by genetics information. And in fact, the DNA is the language of life. Is the DNA that makes you how you are. If you are intelligent, is because of your DNA. If you inherit some of the characters from your parent, is because of the DNA. If you are tall, it's because of your DNA, and tallness is a character that can be inherited from the parent. So therefore, the DNA is a very important molecules that found in every living organisms. In fact, all the characters, all the traits that we have, we actually get them from our parents through the DNA. And this DNA, it has various levels of structure that contribute to its functionality. So there are different levels of protein structure, and it's these levels of protein structure that actually make the DNA to actually undergo so many biological functions in the body. So each level of structure make the DNA to have a unique functionality. So understanding this structure is essential for studying genetics and molecular biology. So if you are able to understand the DNA structure, it's very essential in studying genetics and molecular biology. And so many of you, when we said you don't understand the concept of genetics, so you said genetics is actually a study of variation and heredity. So heredity is when we inherit a character directly from our parent. And of course, when we said variation, it means that the differences that exist between us and our parent. And then molecular biology is a branch of biology that trying to study the informational molecules. And these informational molecules that are very important in producing biotechnology products. So examples of the informational macromolecules that are studied under molecular biology include proteins, uh, DNA, and RNA. They are the informational macromolecules that we studied in molecular biology. So then let's start with the primary structure of DNA. So the primary structure of DNA is referred to as a linear sequence of the nucleotide. So when you have the linear sequence of the nucleotides, then that is when we have the primary structure. And this linear sequence of the nucleotides are held together by phosphodiester bonds. So each of the nucleotides joins with one another through phosphodiester bond. And what are the components of these nucleotides? So the component of nucleotides are one nitrogenous bases, we have phosphate group and sugar. So the nitrogenous bases that are found only in DNA, that we have adenine, we have cytosine, we have guanine, and thymine. So these are the four nitrogenous bases that are found in DNA. So see here, this is a typical example of how thymine and adenine linked together. You see, this is how some of this. So the specific sequence of these nucleotides encode genetics information. So we are coming to that soon. So each of these nucleotides, we have what we call a triplet codons. We have what we call genetic codons. So each of these genetic codes are responsible for coding a specific amino acid. So we use these nucleotides to generate or to encode different amino acids which are used in protein synthesis. So then, what are actually the secondary structure of DNA? So the secondary structure of DNA is characterized by the formation of double helix. So you can see clearly here, this is the double helix structure of DNA. If you can see here, you can see that the DNA is double-stranded. So each of the strands is held together by hydrogen bonding. So DNA form complementary, or hydrogen bonds are formed between complementary bases, where adenine pair with thymine and then guanine pair with cytosine. So this double structure nature, or this complementary nature of the DNA, is stabilized by the hydrogen bonding, and that is what make the DNA to be in secondary structure. So these structures was famously described by Watson and Crick in the year 1957. So you see, ladies and gentlemen, in the primary structure is when you have the linear chain of the DNA through phosphodiester bond, while in the case of secondary structure, the secondary structure of of DNA is formed as a result of complementary bases that joined or stabilized by the hydrogen bonding. So this is a typical examples of secondary structure. So the secondary structure is due to complementary or complementarity of the DNA strands through hydrogen bonding. So this is what make the primary structure differ from secondary structure. So in secondary structure, there is both ester bond and hydrogen bond that stabilize the double helix, while in the primary structure is only the sequence of the nucleotides. So then the next thing is the tertiary structure of the protein. So the tertiary structure of the protein, it involves the three-dimensional arrangement of DNA molecules. So when you have the three-dimensional arrangement of DNA molecules, that is when you have the tertiary structure of DNA. So DNA is further organized into higher order structure through supercoiling and wrapping around histone proteins. So in the primary structure, you just only have the hydrogen bonding that's actually join the two complements, or the two strand of the DNA. So, but in tertiary structure, the DNA further organize itself through coiling and wrapping around histone protein. So the supercoiling and wrapping the DNA around the histone protein is what actually leads to the formation of tertiary structure of DNA. So the three-dimensional structure of the DNA molecules is due to coiling and supercoiling and wrapping around the protein. So this level of structure is crucial for packaging DNA into chromosomes within the nucleus. So we know that, ladies and gentlemen, that if we actually look at how the nature of the DNA looks like, it is a very long molecules, very long and very thin. So generally, if the DNA is not coiled, super coiled and wrapped around the histone protein, it cannot perfectly fit into the chromosomes and into the nucleus of the cell. So that is why in tertiary structure, the DNA super coiled and wrap around the proteins, which make it very tiny and very small, and then fit into the chromosomes within the nucleus. So the tertiary structure is due to supercoiling and wrapping around proteins. So then the next thing is the quaternary structure of DNA. So the quaternary structure of DNA pertains to the interaction of DNA with proteins and other molecules. So you see, in the other one is just coiling and wrapping up the DNA around histone protein, but in the case of high quaternary structure, is the wrapping or is the interaction of DNA with proteins and even other non-protein components. So examples of the proteins that actually interact with this DNA include histone. So histones, for instance, play a significant role in DNA packaging and regulation. So these interactions have formed nucleosomes, which further compact DNA into chromosomes. You see, the tertiary structure locked and supercoiled the DNA and make it fit into the nucleus. But in the case of quaternary structure, the interactions further increase and make the DNA more compact and fits into the chromosome. So in a quaternary structure, a nucleosomes are generated, or a nucleosomes is generated. So that is what we have in the case of quaternary structure. So these are the full level of proteins of DNA structure, starting from primary, secondary, tertiary, and quaternary structure. So then the next thing that we are going to look at is the different types of the DNA. So DNA is classified into various types based on its function and structure. So the most common forms are aDNA and bDNA, each with distinct helical helix. There is also a zDNA, which has a left-handed twists and is thought to play roles in gene regulation. So these are the different types of DNA. So actually we have three different types of DNA. We have aDNA, we have bDNA, and then we have zDNA. So the zDNA is left-handed twisted and it plays a crucial role in gene regulation. So we are going to see them one after the other. And ladies and gentlemen, this is just a structure that trying to show the different levels of protein structure, starting from the nitrogenous bases. So the nitrogenous bases are here. This is their structure. This is how they look like. They are heterocyclic compounds. So they are the nitrogen containing heterocyclic compound. We have adenine, thymine, guanine, and cytosine. And this these nitrogenous bases form or joined together to form the complete nucleotides, and these nucleotides form a complement with one another to form a complete double helix DNA, and then the DNA undergo what we call a central dogma of molecular biology. The DNA converts itself into RNA through transcription, and then the RNA converts itself into protein through translation. So this is what we have briefly on this. So then let's look at the aDNA. So in the aDNA is a right-handed helix that is more compact than bDNA. So you see, all of them, they are right-handed DNA molecules, but the aDNA is more compact, is more twisted than the bDNA. So it typically forms under dehydrated conditions. So it forms under dehydrated condition. That is when there is low water in the environment, and it's not the predominant form of DNA that is found in living organisms. So it's not actually the most predominant DNA that found inside the living organisms. So aDNA is thought to be important in certain DNA protein interactions. So you see, the aDNA is important in DNA, RNA, sorry, DNA protein interaction. So DNA interact with DNA, protein interact with protein, and then DNA also interact with protein. So the aDNA is important in DNA protein interactions. So this is how we have look at it here. This is you see find the right-handed DNA helix. So you see, you have A, B here, and C. So which one is the right-handed DNA? So the right-handed DNA is the aDNA. So then the bDNA. So what is bDNA? So a bDNA is the most common form of DNA found in living organisms. So most of the DNA that found in us, most of the DNA that found in every living organisms, they are in the form of bDNA. So the DNA in Mukhtar Ali, the DNA in Aisha, the DNA in Bashir, the DNA in dogs, the DNA in goats, the DNA of each living organism, bacteria, fungi, they are all in the B form. So it is puces are right-handed helical structure with about 10.5 base pairs per ton. So you see, 10.5 base pairs per ton. So that is why here we say that aDNA is right. So it is more compacted. So it is more compact. It means that in the number of ton, the number of turn per base pair in aDNA is less than the number of turn in vDNA. So that is why aDNA is more compacted than the vDNA. Look at it. The base pair for turn is 10.5 base pair. So it's huge. It's huge. Then this form is biologically significant as it provides the framework for replication and transcription. So this DNA is very important in providing a template for replication and transcription of the DNA molecules. And of course, for the aDNA, it's actually assists or help in DNA protein interaction. So understand the function of each of these two or of each different types of DNA. DNA protein interaction for A, then the B provides a template for DNA for replication and transcription. So then the next one is a zDNA. So zDNA is a left-handed helical form of DNA that can form under certain conditions. So it forms under certain conditions, like in the case of in the case of aDNA, which form under dehydration condition, as you can see here. So zDNA is formed also under some certain condition. So it is a zigzag backbone. It has a zigzag backbone and is thought to play a role. So you see the role of this one, it plays a role in gene regulation and gene expression. So it plays a role in gene regulation and gene expression. So it is significant, is still being studied, particularly in relation to chromatin structure. So ladies and gentlemen, these are the different types of DNA that are actually found in organisms. So they, in conclusion, the different structures and the types of DNA are fundamental to understanding genetics. So each level of DNA structure contributes uniquely to its overall function and stability. So ongoing research continues to unveil the complexities of DNA and its interaction with other biomolecules. So ladies and gentlemen, thank you very much for watching this of my video. If this is the first time you are coming to my YouTube channel, please subscribe and join the YouTube community.